So as I said, the focus of today's first session will be looking at finding features that regulate gene expression. So what I want to do in this first part is to obviously give you a presentation just to explain the data that we have in Ensembl, the sources of the raw data that we use to actually annotate these regulatory features, and then we'll move on to a demo showing you how you can use the Ensembl genome browser to access information and data about those features. So in Ensembl, we annotate regulatory features across the genome. So we call them functional regulatory elements, and they might be things such as promoters, enhancers, or repressors, for example. And we use a variety of different underlying data to annotate these regulatory features. So we look at things such as epigenetic marks, which can include histomodifications and DNA methylation patterns. We also look at transcription factor binding patterns and RNA polymerase binding patterns. And finally, we also look at the pattern of open and closed chromatin across the genome to also help to predict these regulatory features. So the importance of the importance of the regulation of gene expression can be exemplified through the hematopoietic stem cell lineage. If we think about all of the different blood types that exist in adult blood, we can think of all of the different white blood cell types, red blood cells, for example, that exist and have all been derived from a single pluripotent progenitor cell. So it can be deduced that all of the cells within the blood have the same genome. They all are derived from the same progenitor cell. But when we look at them under a microscope, we can see that they all have very distinct phenotypes, very distinct morphologies, and very distinct functions. So obviously they all have the same genome, but the difference in their terminal differentiation that we can see will be the result of different gene expression profiles. So what we know is that in each of these different cells, the genes that are encoded within the genome will be expressed at different levels at different times. And that's all under the control of these functional regulatory elements such as promoters and enhancers. So there are many different layers of transcriptional regulation that can control the expression of a single gene. And I try to summarize this by using this image that is quite well known, and I think it's been used in many presentations and course in textbooks. But I just want to try and highlight some of the main features that I think we can think about when thinking about gene expression. So we might have a gene here, for example, that's obviously encoded, a gene here, for example, that's encoded in the DNA. And immediately upstream of that gene, we might have a promoter region. Now, the promoters, they bind a subset of proteins called transcription factors. And those transcription factors might either directly bind to the DNA itself or bind to one another to form a transcription factor complex. Now, that transcription factor complex will recruit RNA polymerase, which is the machine that actually transcribes the mRNA itself. And so it's obviously the importance of the sequence of the DNA within the promoter region that is responsible for recruiting these proteins. More distantly related from the promoter are other regulatory features such as enhancers. And enhancers bind their own subset of transcription factors. So again, the transcription factor proteins, they will have a motif that they specifically bind within the DNA that will be found within the enhancers. And then the enhancers, the proteins bound to the enhancers come together with the transcription factor complex bound at the promoter in this 3D reconfirmation of the DNA to bring all of these transcription factor proteins together. And it's actually, in fact, this complex of transcription factors bound to both the promoter and the enhancers that result in the recruitment of RNA polymerase and the activation of transcription. There are other features along the genome that also bind different groups. So we can think of perhaps CTCF binding sites within the genome, which can regulate gene expression. And we can also think about methyl groups as well that actually are covalently bound to the DNA sequence. So we know of these repeat regions within the genome called CPG islands, which are stretches of CG repeats with CG bases within the DNA. And the cytosines within these CPG islands can be modified covalently with methyl groups. So an enzyme called methyltransferase can move along the genome and add methyl groups to these CPG islands. And the addition of methyl groups to the CPG islands actually might prevent the recruitment of the transcription factors to that particular regulatory feature. So normally the motif within the DNA sequence that the transcription factor will bind to won't be bound to and recognized by the transcription factor when the methyl groups are there present and blocking the motif from protein binding. So the recruitment of the proteins, the transcription factors to the regulatory features such as the promoters and the enhancers can be determined by a number of different factors. So we've already thought just very briefly about DNA methylation. So this is also exemplified in this part of the diagram here. So we've got this methyl group being covalently attached to a cytosine within the CPG islands. But just to summarize, over on the right-hand side, there are, as I said, a number of different factors that can influence the binding of transcription factors to the DNA. So we can firstly think about the chromatin conformation. So obviously in this diagram, we have a chromosome, which is obviously formed up of this chromatin being wound around and condensed into the chromosome. And this is obviously a diagram that shows this occurring in a very uniform manner, but in fact the condensation of the chromatin is actually a very non-uniform process. And what we actually find is that there are regions of the chromatin that are very densely packed in, like we might see here, and this is associated with transcriptional inactivity. So where the chromatin is tightly wound and condensed, the transcription factors are less able to access the DNA motifs within the sequence, within the DNA sequence, the motifs within the DNA sequence. But there might also be regions that are less well packed in, so the chromatin might be much more open and accessible for the transcription factors to bind to their respective motifs. One of the other things that's very important that we can think about in terms of the accessibility and the binding of proteins to their transcription, the transcription factor proteins to their DNA motifs, is the positioning and modification of the histone proteins themselves. So DNA, as part of its packing and condensation, is wound around an octamer of proteins called the histones. So it's actually 4 different homodimers you can see in this diagram here. So here we just have a front-on view, so we can see 4 of them, but behind there will be another 4. So we'll have 2 copies of histone H2A, 2 copies of histone H2B, 2 copies of histone H3, and 2 copies of histone H4. And these proteins obviously form the nucleosome that these histone proteins will have the DNA wound around them to condense the chromatin. So what we actually know is that the proteins, the histone proteins themselves, are quite globular, but they have an N-terminal tail which extends away from the protein itself. And these N-terminal tails of the histones can be post-translationally modified with a number of different post-translational modification groups, such as acetylation, methylation, ubiquitination, and sumorylation. And these different modifications can influence the recruitment of transcription factors to specific motifs and therefore affect the transcriptional activity. So there are a wide array of experimental techniques that we as scientists can use to explore the different levels of transcriptional regulation that is present across the different regions of the genome. So again, this image is taken from the ENCO project, you can see, and it tries to describe the different ways that we can assay the transcriptional regulation at these different levels. So firstly, we have the chromosome conformation and the chromosome conformation, which we can see through combinations of 5C, GeoPet, and Hi-C experiments. Then we can have a look at regions of open and closed chromatin, so whether they're open or closed, like we might see here. And we can look at that using techniques such as DNA-seq, FAIR-seq, and ATAC-seq. The next thing that we can do is we can look at a number of different things with an experiment called ChIP-seq. So this is chromatin immunoprecipitation followed by sequencing. And what this does is it allows us to look at the proteins that are interacting with the DNA. So these proteins might be the transcription factors themselves. So using ChIP-seq, we can experimentally assay to see where along the genome the different transcription factors are binding. But we can also use ChIP-seq to interrogate the different histomodifications that we can observe across the different regions of the genome as well. So we can use antibodies in our ChIP-seq experiment that bind specifically to histones that are modified in a particular way, and use that followed by sequencing to find the type of histone modification that we observe in a given part of the genome. We can look at things like methylation, so DNA methylation, by using whole genome bisulfite sequencing or reduced representation bisulfite sequencing or methyl arrays. Then we can look at the gene level, the gene expression itself. So we can look at computational predictions, we can look at RNA-seq data, and we can actually look at the transcriptional efficiency by using ClipSeq and RipSeq as well. So this is just a summary to show you that there is a wide array of experimental techniques to interrogate the transcriptional regulation that's occurring across the genome. So what I want to do now is think about how Ensembl can make use of the data from all of these different types of experiments that's being generated by consortium and groups all around the world, and use that to annotate regulatory features across the genome. So what I want to do is go through the different steps of the Ensembl regulatory build, and then we'll think about these in a bit more detail in the following slides. So in step one, we take raw data from a number of different sources. So these different collaborative projects have looked at a number of different cell types for human and mouse. And we take that raw data from these different experiments for the different cell types. The step two is to then process this raw data, align it to the genome to predict the positions of the regulatory features. And we assign the regulatory feature types such as promoters, promoter flanking regions, enhancers, CTCF binding sites, open chromatin regions, and transcription factor binding sites as well. The third step is to then predict the activity of these features in the different cell types that we have experimental data for, available for. So the activity that we assign might be active, poised, repressed, inactive, or unknown. So for each cell type, we predict whether that regulatory feature is active or inactive in those different cell types. And then finally, we display all of this information through the genome browser. And we're going to see how we can get that in the demo. So firstly, I want to explain where all the data comes from. As I've said, for the regulatory build, we only produce this data at the moment for human and mouse. These are the only two species that have experimental data for cell types that we can use. As you'll see at the end of the presentation, we do have long-term plans to extend this annotation to other species as well. But at the moment, just human and mouse. So for human, there are three main projects that we have experimental data for. So the first is the ENCODE project, and the second is the Roadmap Epigenomics project. So both of these projects looked at cultured cell lines. So the ENCODE project looked at immortalized cell lines, and the Roadmap Epigenomics looked at primary cultured cell types. So for these 48 cultured cell types in human, from these two projects, we have ChIP-seq data, transcription factor binding site data, RNA polymerase data, and DNA sensitivity data, so the open chromatin patterns. For mouse, we have something similar from the ENCODE project for 8 different immortalized mouse cell lines. So we have, again, ChIP-seq, transcription factor binding site, RNA polymerase, and DNA sensitivity data for these 8 mouse cell lines. And then finally, for human, we also have data from a third project called the Blueprint project. And this actually looked at 20 different, or actually now more, primary cell types from the hematopoietic cell lineage, so direct from human cells. So the Blueprint project looked at ChIP-seq data and DNA sensitivity data across these different blood cell types. So as I said, the step one is to take this raw data from all of these different sources, align it to the genome, and then perform a process that we call peak calling. So what we can see here in blue is a representation of the genome itself. And then the data is represented as these colored lines. So the alignment of this data to the genome is represented as these traces, or what we call signals. So we can see, for example, here that we have this red signal, which is sort of quite low as we move along the genome. And then as we get to this region of the genome here, the signal passes through a peak, and then it returns to its baseline level. And we do the same thing for all of the different experimental data types. So we align them all to the genome, and we look to see where the signal for each of these experimental types passes a particular threshold. So here we can see the red and the purple data cell type have passed a particular threshold. So we label these as peaks. So you can see these distinct and discrete blocks at the top here, these colored blocks. These are what we call peaks that correspond to where the signal passes a particular threshold. And we can do the same all along the genome. You can see here this pink block comes from this increased signal. And then we have this actual grouping of peaks that are all correspond to signals that have increased in this region of the genome. What we then do is we look to see whether there are different patterns of signals and peaks that all correspond to a single region within the genome. Like you can see here, there's a cluster of peaks all in the same region. And we look across the genome. So then within step 2, we look across the genome to see if there are corresponding signature patterns, peak signature patterns across the genome. So you can see here, for example, all of these regions of the genome have the same signature of peaks. So this transcription factor B and transcription factor C and histone modification 1, 2, and 3, they all occur at these different places within the genome. And if this particular signature of transcription factor binding and histone modification, for example, is a known promoter, then we can infer that everywhere else in the genome that we see this same signature is also a promoter region. And we do the same things for different signatures. So we look for different evidence signatures, and if there's experimental evidence indicating that those regions are regulatory features, then we also annotate those across the genome. The second part of step 2 is a process called segmentation. So this is where the different regulatory features are categorized into different groups based on the underlying signatures, so the underlying experimental data. So we can see here, for example, we have one type of feature based on the underlying signature. So this might be a promoter, for example, and then we have a different signature here with different evidence types underneath, giving us another type of regulatory feature. So this might be an enhancer. So we give the features, we categorize the features into different subtypes based on the type of underlying data that we have for each feature. Step 3 is to then predict the activity of these features in the different cell types that we have available in Ensembl. So what we do is we take the regulatory feature annotation across the genome for all of the cell types. We're just using 4 here as an example. And then what we do is we move along the genome in 200 base pair blocks, asking at that position, is there any transcription, are there any regulatory features annotated? So in this case, we would say yes. So in cell type 4, part of this 200 base pair block has a regulatory feature annotated. So we annotate a regulatory feature here. And we move along the genome in these 200 base pair blocks, asking the same question. So again, the answer would be yes and yes. And moving along the genome, asking the same question. So here we can see now another feature annotated based on cell types 1, 3, and 4. And then finally, we have our final feature. So what we're actually doing at the top here is constructing our regulatory build. So this is our multi-cell regulatory features. And then the final step is to look in each of the cell types again and ask for each of those cell types, is there any evidence to suggest that those regulatory features are active, poised, repressed, or inactive? So in this case, for example, we can see that in cell types 1 and 4, there's evidence to suggest that these regulatory features are active, so they'd be colored in red in this case, whereas in cell types 2 and 3, there's no evidence to suggest that they are inactive or poised or actively repressed. So they are inactive, which is what we can see depicted here. And we'll see in the demo how we can look at the regulatory feature activity in different cell lines in the browser. So step 4 is to view all this data in the Genome browser, which is what we're going to do in a minute. We're going to look for different cell types. We're going to look for the regulatory features and their activity within those different cell types. So before we start our demo, it's very important for me to tell you what Ensembl does not do. So what we do not do is link regulatory features to genes. What we have is a single reference genome sequence that we annotate genes on. And then against that same reference genome sequence, we annotate regulatory features. And what you can do is you can look at the reference genome and look at both the gene and the regulatory feature annotation and look to see where those features might coincide. So you're able to perhaps look to see where they coincide. You might be able to make some correlations between expression level patterns in different tissues and the activity of regulatory features in different cell lines. But really the take-home message is that you're required to make your own inferences about this data. So if I'm looking at the genome, at a gene of interest, and I can see a promoter region annotated right at the 5' end of my gene of interest, I might be able to make a good guess that that is a promoter that controls the expression of my gene of interest. But that's an inference that I'm making as a scientist looking at the data, which I would then need to experimentally validate and verify. So there are obviously limitations to the data that you're looking at in Ensembl. And this is because the data is actually really, really complex. And we're still, well, as with all the other annotation as well, it's always going to be refined and the annotation will be improved. So we do hope to eventually, at some point in the future, be able to link regulatory features to the genes that they control the expression of. So some of the other regulatory data resources that we have in Ensembl, firstly for human and mouse, we have Vista elements. So the enhancers that come from the Vista project, we have annotated in Ensembl, so you can see those and view those in the browser. We also have predictions of mRNA targets from Tarbase. And then for human only, we also have the phantom transcription start sites and enhancer annotation, and also the GTEx eQTL data, which I showed you very briefly yesterday. Finally, we also have methylation data from the ENCODE project that you can add as tracks to the location view. And within the gene and transcript, within the transcript tabs, you can also view the microarray probe mapping to the different transcripts of your interest. As I said, in the future, what we hope to do is to extend this technology to a number of different species. So there are a number of projects that are aiming to generate similar types of data for different cell lines, for different species. So the FANG project is looking at agricultural species. And there's other projects such as AquaFang looking at fish species, and then there's also the zebrafish genome biology consortium that's looking at getting similar data for the zebrafish model organism. And obviously, as I said, one of the major goals of our regulatory build is to eventually be able to link the regulatory features to the genes that they control the expression of. So some of the data that we're looking at using to try and make these associations is firstly EQTL data and also high-C data that is currently being produced, published, and made publicly available, which we'll be able to then import and use in our own annotation process. Good. So what I want to do in the demo is look at a region of look at a genomic region around a gene of interest. So BPTF will be my gene of interest for this example. And what we're going to do is we're going to look at the regulatory features that fall around my gene of interest. And then I'm going to pick on one of those regulatory features and explore it in more detail. So if you're following along with me, the screenshots start on page 46 of the course book. Or you can follow along with me as well. So in your internet browser, you just need to open up a new tab and go to the Ensembl homepage. And from the homepage, you just need to search for your gene of interest. So just as we did yesterday and the day before, we can type our gene name, BPTF, and then we can click Go. And then you've got a couple of different options here. So we have the link for the BPTF gene. So this will take us to the Gene tab. Underneath, you can actually see a link for the location where this gene exists. So you can actually directly click on these coordinates within the description. So you can click here, or you can click on the gene name and then toggle across to the Gene tab, the Location tab like we've done before. So I'll click on the gene name, but then from the Gene tab, I'll directly click on the Location tab to take me to the location view where this BPTF gene can be found. So it's on chromosome 17, you can see. So we can see our location view. In fact, we've got lots of tracks added from all of our previous demos. You can see all of the conservation scores, for example, and the variation tracks. But the track that I want to draw your attention to is here. It's called the regulatory build. So this is what we call the multi-cell regulatory features. So these are the features that have been annotated across all of the different cell types. And the colors of the blocks, as we've sort of already discussed in the presentation, represent the type of regulatory feature. So if we scroll to the bottom, we can see our regulation legend. So we have this dark red is the promoter regions. The yellow gold color is our enhancers, where we have the salmon color, it's the promoter flank. CTCF is the sort of turquoise color, and then we have open chromatin in gray and transcription factor binding sites in this light purple color. So we can begin to actually see this now across our region of interest, and we can correspond that with the start and end of our gene. So we can see here, this is the BPTF gene. We can see this is the start of our gene. This is the most 5' exon. We can see the direction of transcription. So this is the 5' end of our transcripts of our gene. And we can see that actually they fall in the same region of the genome where we see this promoter, so this dark red region. Now, obviously, this actually is right at the end of where our view exists. So what I'm going to do is I'm just going to click on the double-headed arrow at the top here, and then click and drag the genome, just so that I get a bit of a better view of the 5' region of the gene and some upstream sequence as well. Good. So now we can see the full length of the promoter region where the 5' exon of the transcripts of the BPTF gene can be found. And then we also see the surrounding features as well. So we have a number of enhancers here, and then also some promoter flanking regions and CTCF binding sites as well. So if you want to find out more information about a regulatory feature, so if I'm looking at this promoter here and I've become interested in it because it's falling at the end of my gene of interest, at the beginning of my gene of interest rather, I can click on it within the view. And then from the pop-up window, we can see the stable ID of the promoter. We can see the type, so it's a promoter, we know that from the coloring, and then just some information about its genomic location. So to get more information, we need to click on the stable ID. And that will take us to the regulation tab. So what we can see now is that we've obviously got the location and the gene tab that we originally searched for, but we've now opened up this new tab, the regulation tab, that will have all of the information about our regulatory feature of interest. So what we can see is that we have landed on a summary page from the menu on the left. And within the summary page, there's a number of different bits of information we can see. So firstly, we can see the classification, so we know it's a promoter. We've got the location of it as well. And then in the image underneath, we can see some different tracks with different types of data. So we've got the gene that we originally searched for, and then the regulatory build track. So we have the annotation of the promoter. Then we have data from the Phantom5 project. So transcription start sites and enhancer predictions we can see here from the Phantom5 project. Then we also have mRNA target predictions from Tarbase. And then we also have motif features. So what we can see here, this is obviously quite a large track. There's lots of data here, but these are essentially the DNA motifs that are predicted across the genome. Now, the black motifs are experimentally verified, whereas the gray motifs are predicted. So to get more information, we can click on one of them. So we can see here, for example, the motif feature. So it has its own ENSM number for Ensembl motif. We have the location, and you can also see the transcription factors that are bound to this motif feature, so LEF1 and TCF7L1. And this binding has been experimentally verified in K562 cell line. If we want to see the binding matrix, we can actually click on the link for the binding matrix within the pop-up window. And we can see that here. So you can then download that. So we can see it in the browser. So you've got options to view the frequency or the bits. And then as well, you can download the image as a CSV, or you can download the data as well. So you can download the image or the data. At the very bottom of the page, we can see there's a table that shows us the information about the activity of this regulatory feature in the different cell types that we have in Ensembl. So we can see that in 6 of the 118 cell lines that we have in Ensembl, this promoter is active. So these are the different cell lines where this promoter is active. Then we have a number of different cell lines where this promoter is inactive. 98 cell lines we can see here. They're all listed. There's one cell line where it's being predicted as poised. And then there are 13 cell lines where this cell type is predicted to be actively repressed. So there are transcription histone modifications perhaps, and DNA, there's transcription histone modifications that might be actively contributing to the inactivity of the promoter in these cell lines. If we scroll back up to the top of the page, we can begin to find some more information about the underlying data for this regulatory feature in the different cell lines. So the first thing I'm going to click on here is the icon at the top called Details by Cell Type. And what we can see here when we first arrive at the page is a summary view, again, with the regulatory feature with the gene of interest. So this is something similar to what we saw on the previous page. But what we can do now is click on this blue button, Configure Cell/Tissue, which will allow us to configure the page to show data for our cell types of interest. So from the pop-up window, we're taken to quite a complicated display because of the number of different cell lines we have, that allows us to select the data of interest. So the first thing to do is click on the flow chart at the top. So we click on Select Tracks. So what we want to do is to select the cells and the experiments that we are interested in. So firstly, we can click on Cell/Tissue, and then you can either find them by searching in the box here, or you can scroll using this alphabetical list to find the cell lines of interest. So the cell lines that I'm going to use are the GM12878 cells. So you can click on G and then click on the GM12878 box to turn that cell line on. And I'm also going to choose the HUVEC cell. So you can click on H and then select HUVEC as your cell type. So as you click on different cell types, you can see over on the right-hand side that the number of cell types that you've selected will be displayed. So at the moment, I've got 3, but if I turn off here these HUE S64 cells, I'm back down to 2. So what I'm going to do in this demo is just compare the HUVEC with the GM12878 cells, but you can turn on as many cell lines as you like. Obviously, the more cell lines you turn on, the more data that you're trying to view, and it will take a long time to load. So I just want to use 2 as an example here. The next thing to do is to select your experiment, so the experimental data that you want to view. So you can click on Experiments in this window. And the experiments fall in 3 different categories. So histone, open chromatin, and transcription factors. So we can see here, by default, I have all of the histones, all of the histone data selected, but you can click Deselect All. You can select specific histone modifications that you're interested in, or you can click Select All, so that you can now see that I'm viewing 27 out of 27 available histone modification data types. Then I can click on Open Chromatin, and I can see I'm also going to view the DNase one. And then for the transcription factors, we can see that there are, again, a number of transcription factors that we can search for alphabetically. I'm looking at 2 by default, but you can turn on as many as you like, or you can turn on Select All. Again, I'm just going to select a few for the time being, because selecting all will mean that the demo takes perhaps too long to load. So just select a few for the purposes of the demonstration. And then the next thing to do is to configure the track display. So now we've selected the data that we want to see. Now we just want to choose the formatting. So we can click on the next part of our flowchart, Configure Track Display. And now you can see this chart with the boxes that allows us to select our formatting. So for the 2 different cell types that we have chosen to see, we can click on the box to see if we want to see the column and the cell, and we can turn them on. So that we can, for the epigenomic activity, and the segmentation feature, we can turn them on or off. And then for the different experiments, we can choose whether we see the peak and the signal, or the peak only, or the signal only. So I'm going to choose peaks and signal for all of my experimental data types that I'm viewing. Now, it's important to notice that for some cell lines, you can see that there are some white boxes. So here, the histone 3 lysine 4 dimethylation pattern is not available as a data type for the GM12878 cell. So this just means that for this cell line, this experimental data is not available. So I'm going to show it for the HUVEC cells, but it's not going to be displayed for the GM12878 cells because they're not, the data is not available. So once I'm happy, I can click on View Tracks. So we've got the regulatory builds here for the multi-cell feature. Then we have a section all relating to the GM12878 cells. We've got the activity, which is depicted by the colored block, the colored feature. So we can click on the feature and we can see that it's a promoter and its status is active. And we can see all of the different motif features that fall within this regulatory feature. And again, we've got the binding matrices as well. Then you have, for the GM12878 cells, you've got the signal running across the bottom. And then you have the peaks where each of these signals passes the baseline threshold. So we can see perhaps, as an example, for the TAF1 transcription factor, we can see this light blue signal has passed this peak here. The signal has passed a threshold. So we have this TAF1 peak indicating that there's a TAF1 transcription factor binding at this region in the genome. The same for all of the other experimental data as well. Then for the HUVEC cells, we have a whole section relating to the HUVEC cells. So we have the regulatory feature, which is gray. And when we click on the feature itself, again, a pop-up window will show us the name, the ID of the feature and its status, which will be inactive. So we can see, okay, so sorry, I actually misspoke. So this feature is repressed. So the gray color shows that this feature is repressed. And so what we can see is, again, when we look at the signal, we have these experimental data, and where it passes the particular threshold, we call these peaks corresponding to the data itself. So what we can see is that these, well, there's not really any transcription factor binding in this region, but there is a number of different histomodifications that might be associated with the repression of this regulatory feature. Good. So as I was just going to mention before the page loaded, I think Emily's been mentioning about the source of the data. So you can view the sources of the data by clicking on the source data button at the top here. Or also you can go to the help and documentation that's linked at the blue bar at the top and navigate to the regulation section to get the information about the projects, the cell types, and all of the data types. So that brings me to the end of the demo. Thank you for your patience while the page was loading, but I'm glad that we did persevere because we managed to see the data within the browser itself, showing you how you can explore it and view that. So what I would like to do now is give you an opportunity to have a go at the exercises for yourself. So what I would advise you to do is navigate to the question book where the exercises can be found. So page 14 for the regulatory exercises. So all we have to do is open the question book and scroll down to page 14. And that's where you'll find the exercises for the regulation section here. So there's just two exercises looking at human enhancers and then also a human regulatory feature as well. So you can search for them using the stable IDs or using the by looking in the location tab using the gene names as well, like we did in the demo.